So this is lecture 2 in the course on principles of metabolism. And in this video lecture, we'll look a bit at the materials and energy that a cell consumes and produces. I like to think about this kind of as a budget for the cell in numbers, how much substrates are taking up, how much products are produced, and what is the real energy turnover of a cell. So this is still a fairly big picture topic, but I think it's useful if you're starting to study a new cell type perhaps, and you want to get a first sense of how metabolically active is it, and what are the major processes in this cell type. How much oxidation does it do? How glycolytic is it? What are the major processes of producing biomass, and so on. So let's look a bit at the some kind of typical values. It's hard to say specifically values for any cell type. Of course, they vary. These values vary a lot across cell types. But let's look at some reasonable range and try to get a sense for what these numbers could be. So let's start with energy metabolism, and let's start looking at uptake of nutrients. So of course, different cell types have different preferences for what nutrients they use, but a useful summary measure can be to think about how much carbon they can take up, because this is really the major determinant for energy metabolism, and it determines biomass also to a large extent. So how to measure this? So I tend to use a unit of femtomolar amounts per cell and hour. It's a reasonably sort of useful unit. You have reasonable numbers in those units. And molar amounts are usually good if you are in the business of calculating molecules. And so, for example, if you are thinking of glycolysis and you want to know how many lactate you get from glucose, you get 2 lactate from 1 glucose, so you want to count molecules. And then molar amounts can be useful. Or if you're trying to figure out how much oxygen a cell would consume based on the nutrients it consumes and so on, then molar amounts are usually good. Sometimes it's a bit hard to understand though. So here I'm saying that a cell takes up about 203,000 femtomolar. So what does that really mean? We try to get a sense for it. So it can be useful to convert it to mass. So then we get to 40 to 50 picograms, and that you can compare to the weight of a cell. And so how much is this? Well, as you see, it's a large range. The lower numbers are typically for oxidative cells. They require less carbon because they use it more efficiently. The higher range is for a very glycolytic cell. And so a glycolytic cell will waste a lot of carbon and therefore it uses a lot. But how much is it exactly? Well, if you think about a glycolytic cell that consumes a lot of glucose and spits out a lot of lactate, a very glycolytic cell can consume its own weight in less than a day, maybe 12 hours, depends a bit. So that's how much these numbers mean, and it's a lot. Cells can really sort of process their own weight very quickly in this mode of metabolism. So if you think about respiratory cells, it's a bit different. So then the lower numbers apply for nutrients. And you can calculate how much oxygen a cell consumes by looking at, there are established ratios of how much oxygen you need to oxidize a specific amount of nutrients. Or you can actually measure it. I mean, there are measurement devices which are fairly good for oxygen. Some of the other measurements here, by the way, we determined in a paper we published in 2012, when we did a lot of quantification of what cells take up and release. If you know oxygen, you can also determine CO2. So you can say a bit about how much of the carbon is actually oxidized and leaves the cell as CO2. So there's the concept of the respiratory quotient, which you probably have heard of if you're working more in whole body metabolism. And so you can connect these numbers. And this is what it looks like, up to a couple of hundred femtomolar oxygen and CO2 per cell an hour. So this is much less in terms of material. So if you have a respiratory cell, it might sort of process its own weight in nutrient and in biomass in a couple of weeks, maybe a month. So a respiratory cell can, like if you think about a quiescent cell, which mainly just sits there and derives its energy from respiration, it can subsist for a couple of weeks or maybe up to a month on material the size of itself. And this is, you see in autophagic cells that sort of break down their own content, that they can last for quite some time on their own biomass. So there's a large difference between highly glycolytic cells and respiratory cells. If you are consuming amino acids, if a cell is consuming amino acids, then you have to get rid of the ammonia somehow. And this is also an important flow, I think, an important flux in energy metabolism. And it becomes a practical problem for many cells and a problem for cell culture because it builds up in cell culture, and in a few days you can reach toxic levels of ammonia. So that gives you some sense of how large that flow is, in a sense. So when it comes to ATP, this is also a large flux, a quick turnover. You can count this up to in the thousands of femtomolar per cell an hour. And here I think it's useful to think of turnover time. It's not so useful to think about how much weight of ATP is processed, because this is a cycle and it turns over. But if you think about the pool of ATP available in a cell, the turnover time for a highly energetic cell is in the order of minutes. So that's a good number to know. So if you would block energy metabolism or block ATP synthesis, you will have a crisis in the cell on the order of minutes. So that's interesting to know. It's also interesting to know that the efficiency of actually capturing energy from nutrients into ATP is actually not that good. It might be 30%, might be a bit higher. And so not all the energy that that cells consume and extract is actually captured efficiently as ATP. So there are other kinds of energy in cells than ATP. You can think about energy turnover in terms of actual watts. So if you calculate this, you can get it to a cell is about 50 nanowatts of sort of power, energy turnover. You can probably tell I'm an engineer by background, so I think these kinds of things are kind of cool. But so what is that good to know? Well, it takes a billion cells to make a light bulb. I don't know if that's helpful, but it gives you some sense of how much power we are talking about. What is the rate of energy turnover? Okay, so that's the big picture of energy metabolism. One question that comes up a lot, especially if you look at proliferating cells and especially in cancer metabolism, perhaps, is what fraction of the ATP comes from glycolysis and respiration. These are really the only two quantitatively important processes when it comes to generating ATP in cells. And this has been debated quite a bit because people argue about how important glycolysis is. But surprisingly, this is a fairly simple question, but surprisingly, there is not that much data on it. In order to actually determine this, you have to measure both oxidation, so oxygen consumption, and glycolysis, which is usually lactate production, approximately. You have to measure these things quantitatively in an accurate way and at the same time in the same cell system. And this hasn't been done very often. So this is from a review by Su and Guppy from 2004. It's 10 years old now or more, but I think it's still the latest review of the quantitative data. And most of the data in here is very old. It's from the maybe 70s or 80s. So a lot of numbers here, and it's not super useful to look at all of this. But I would like to focus your attention on this column, which is the fraction of ATP that comes from glycolysis. And the take-home message here is that it varies a lot. So there are definitely cells, these are all normal cells, by the way. There are definitely cells that are very low on glycolysis, so they derive virtually all their energy from respiration. But there are also normal cells, like, well, I don't know, endothelial cells up here, which have been measured to be very glycolytic, to derive most of their energy from glycolysis. So one important thing to know is that glycolysis, which we often talk about in cancer, is not exclusively a cancer phenomenon. There are plenty of normal cells that do glycolysis quite a lot. And if you're studying a new cell type, or starting to study a new cell type, and you don't really know how it behaves, I would not make any assumptions about this until you have actually measured it. So in numbers, where does all the energy from energy metabolism go? So this is also difficult to measure and come up with, and some of these numbers are theoretical, but some of them people have been able to measure. This is also from the review by Rolfe and Brown. They try to come up with a whole body average, sort of take into account roughly the composition of cell types and organs and so on. And so if you do something like that, it looks kind of like this. So first of all, they base this largely on oxygen consumption because it's the major, respiration is the major mode if you think about the body as a whole. And most of the oxygen consumption is mitochondrial. There is some oxygen that goes elsewhere, but we can largely ignore it. And most of the mitochondrial oxygen consumption indeed goes to ATP synthesis, but there is quite a bit of things, of energy that just goes to proton leaks across the mitochondrial membrane. And so this you can think of as a heat, or maybe as a waste. But cells also need to generate heat to keep the temperature. So that's an important part. But then if you look at the energy that actually goes to ATP, that's this part, you can start looking at what processes in the cells are receiving this energy. And protein synthesis is a large fraction, and that probably most people would not be surprised by. But what most people might be surprised by is the fact that this part, the ATPases, the ion gradient pumps, is a very large chunk. It can be larger than protein synthesis. This is also true if you look at individual cells. So this is another is a primary paper that looked at a tumor cell in mouse, also showing that the ATPases are important. Sodium, ATPase, and calcium transport. So these are large shares. So the cell spends a lot of energy actually building up these ion gradients. And you can actually think of it that the cell is storing quite a lot of metabolic energy in these ion gradients. So it's an important sort of reservoir of energy. It's both the plasma membrane and it's also stored in the mitochondrial membrane. So ATP and the pool of ATP in cells is not all of the energy content. There's a substantial part stored in these gradients. So that's important to know, I think. And besides these, there are a few major processes if you look at the body as a whole. Gluconeogenesis and the generation of urea to get rid of nitrogen in the liver are important, and then muscle contractions and so on. But at this point, it gets, of course, very dependent on what cell type you are interested in. But this is a general sort of overview of how energy is produced, where does it come from, and where does it go. So the other aspect is biomass. And if you have a biosynthetic cell or a proliferating cell, this becomes important to know about. So the composition of biomass is pretty much the same in most cells, and you'll find this in any basic biology textbook, obviously. So the volume of a mammalian cell is a few picoliters. You can calculate its dry mass easily. And most of this is protein. A fairly large share is lipids. And then smaller amounts are carbohydrates and nucleic acids. And DNA is actually quite a small fraction of the nucleic acids. So why is that good to know? Well, if you're thinking about cells that proliferate, this determines what they have to build, obviously. So proliferating cells, of course, have to copy themselves. So they have to generate this biomass. And if you know the composition of biomass, you can figure out what they have to synthesize. And that can be quite useful when you study metabolism. You can also figure out how much they synthesize, of course, because you can easily measure proliferation rates. And a rapidly growing cell line divides in 1 day, as I'm sure you know. And so that's the amount of biomass you have to synthesize, a few nanograms a day per cell. So it synthesizes its own weight every day. So that's a fairly fast process. If we think back to the numbers we had for glycolysis, that was also on this order. So a rapidly glycolizing cell will use carbons at this rate. So we can immediately say from this that even if you have something that is very glycolytic, a large fraction of the carbon actually goes to biosynthesis, maybe half. Of course, this is very rough as a number. If you have an oxidative cell that proliferates, actually most of the carbon will go to the proliferative pathways. So then you should expect that most of the nutrients is shunted into pathways for biosynthesis rather than energy production. One major sort of unknown in this space is macromolecules as nutrients. So we know from old biochemistry papers, and there are a few recent contributions in this field, that many cells, at least transformed cell lines, take up macromolecules by endocytosis and processes like that, and use them as nutrients. We don't know how much this is, but all of us who do cell culture-based work grow cells in serum, and serum contains a lot of macromolecules, a lot of protein, but also lipoproteins perhaps. And this is something to be aware of when we are quantifying uptake and release of nutrients and waste products. We don't usually measure this. So this is something of a white spot, I think, in the literature on biosynthesis and these processes. So something to be aware of. But this is pretty much the overall picture. Many of the numbers, again, in this talk are very approximate and roughly calculated. But it's useful to do this for your cell line of interest and get a sense for its size and the most important processes of metabolism.